The findings are being reported in the journal Advanced Functional Materials, in a paper by recent MIT graduate Victor Leon PhD 23, professor of mechanical engineering Kripa Varanasi, former postdoc Baptiste Blanc, and undergraduate student Sophia Sonnert. No matter how successful efforts to reduce or eliminate carbon emissions may be, there will still be excess greenhouse gases that will remain in the atmosphere for centuries to come, continuing to affect global climate, Varanasi points out. The findings are being reported in the journal Advanced Functional Materials, in a paper by recent MIT graduate Victor Leon PhD 23, professor of mechanical engineering Kripa Varanasi, former postdoc Baptiste Blanc, and undergraduate student Sophia Sonnert. No matter how successful efforts to reduce or eliminate carbon emissions may be, there will still be excess greenhouse gases that will remain in the atmosphere for centuries to come, continuing to affect global climate, Varanasi points out. There's already a lot of carbon dioxide there, so we have to look at negative emissions technologies as well, he says, referring to ways of removing the greenhouse gas from the air or oceans, or from their sources before they get released into the air in the first place. When people think of biological approaches to carbon dioxide reduction, the first thought is usually of planting or protecting trees, which are indeed a crucial sink for atmospheric carbon. These algae grow anywhere from 10 to 50 times more quickly than land-based plants, and they can be grown in ponds or tanks that take up only a tenth of the land footprint of terrestrial plants. What's more, the algae themselves can then be a useful product. These algae are rich in proteins, vitamins, and other nutrients, Varanasi says, noting they could produce far more nutritional output per unit of land used than some traditional agricultural crops. If attached to the flue gas output of a coal or gas power plant, algae could not only thrive on the carbon dioxide as a nutrient source, but some of the microalgae species could also consume the associated nitrogen and sulfur oxides present in these emissions. For every 2 or 3 kilograms of CO2, a kilogram of algae could be produced, and these could be used as biofuels, or for omega-3, or food, Varanasi says. Most algae grown commercially are cultivated in shallow ponds, while others are grown in transparent tubes, called photobioreactors. Because the cells naturally carry a small negative electric charge on their membrane surface, the team figured that electrostatic repulsion could be used to push them away. The idea was to create a negative charge on the vessel walls, such that the electric field forces the algae cells away from the walls. Another way to describe it is like a force field, whereas before the cells were touching the surface and getting shocked. The material can be applied in a coating that is vanishingly thin, just 10 to 20 nanometers thick, so very little would be needed to coat a full photobioreactor system. What we are excited about here is that we are able to show that purely from electrostatic interactions, we are able to control cell adhesion, Varanasi says. Additionally, Leon says, since we're using this electrostatic force, we don't really expect it to be cell-specific, and we think there's potential for applying it with other cells than just algae. In future work, we'd like to try using it with mammalian cells, bacteria, yeast, and so on. It could also be used with other valuable types of algae, such as spirulina, that are widely used as food supplements. The same system could be used to either repel or attract cells by just reversing the voltage, depending on the particular application. Instead of algae, a similar setup might be used with human cells to produce artificial organs by producing a scaffold that could be charged to attract the cells into the right configuration, Varanasi suggests.